Hello, I'm Andrew Donner, and over the summer I worked on the in situ study of Alzheimer's disease rat brain tissue through gold colloid aggregate and attempting the detection of amyloid oligomers. I worked on this with Kevin Chung and Dr. Yokoyama. So to start off, some background information is that the brain is severely damaged by Alzheimer's disease with many symptoms including memory loss. In our research, we used the amyloid beta 140 peptide, which is a biomarker associated with Alzheimer's disease. We have put together an approach to detect the formation of amyloid oligomers interacting with gold colloid nanoparticles that were monitored through the surface-enhanced Raman scattering methods. The purpose of this research is to detect amyloid oligomers in AD-positive rat brain tissues through the use of gold aggregates, and this will allow us to gain information about oligomers. So to now look into more of the methods. On the left there, under methods, you can see more of the setup and the preparation that we used. But I'd like to draw your focus to figure 2 and 3, where you can see more of the techniques that we used. We originally use a microscope to look for our gold aggregates, and we take a white light image of the gold blocky shaped pieces that you can see in figure 2. And then after that, we go to figure 3, where you can see the spectrums that we get from our SIRS technology and the two different components that it yields, and those two spectrums right there. The results that we get from using those techniques is that in our data, we found very clear results visually and also through spectroscopy. As you can see in figure 4, we have big differences in the hippocampus regions compared to the other brain samples. In the hippocampus, we get a lot more aggregates, as you can see in the white light images, compared to the clustered characteristics of the non-hippocampus tissues. We also observed in our SIRS spectrums that we collected that there was a lot more signals in the 1000 to 1700 Raman shift range in the hippocampus rather than the non-hippocampus tissues. So these two observations put together lead us to believe that oligomers and AD activity mainly occur in the hippocampus regions of the brain. So in conclusion, this study characterized the distinct features exhibited in AD-positive rat brain tissues before the terminal stage of Alzheimer's disease. This established a sensitive method to identify oligomer aggregates in rat brain tissues, which contain unique SIRS signals, which were mainly observed in the hippocampus regions. Our future research can look into AD-positive rat blood to observe if any aggregates can be found there. So thank you for listening, and it has been an honor to present at this presentation.